Today I'm gonna talk about myogenesis, but from a different angle than we may be used to. And essentially, where we're gonna start is with the classic. Everyone knows the classic progression of myogenesis, but what I'm gonna look into is underlying this is the epigenetics behind how these factors, some of these factors may work, and how does the epigenetic landscape of the cells actually contribute to myogenesis. And we kind of stumbled along this in a unique way. We actually started looking at rhabdomyosarcoma, which is the most common soft tissue sarcoma in children. And these cells are myoblast-like cells. They have a gene expression profile that's similar to myoblasts, but they're caused by, there's two different types. The most common type is called embryonal, and it is activated mostly by mutations in the RAS pathway, MAP kinase pathway, with about 25% of the mutations actually occurring in RAS oncogenes. The other type is actually caused by a fusion gene between either PAX3 or PAX7 and FOXO1, where the binding domain of PAX3 or POX7 is fused to the transactivation domain of FOXO1. And so because this is childhood cancer, there's not a lot of mutations besides some of these oncogenes. And so there's a big component of epigenetics in these cancers. And so one thing that we looked at when we first started looking at these cancer cells is we wanted to see what happens when we take out the effect of the oncogene itself. So here we're looking at the alveolar type that has the oncogene that's the fusion between PAX3 and FOXO1. And if we use CRISPR high efficiency CRISPR to ablate the oncogene, we were surprised to find that the cells don't die, that they actually differentiate into muscle fibers. You can see multinucleated muscle fibers stained by myosin with PAX3 knockout, FOXO1 knockout, and then PAX3 and FOXO. And the nuclei are stained with DAPI. And so you get these large, in this particular cell type, this is arrived from a child. This particular cell type produces huge muscle fibers. In fact, sometimes they can essentially take up the entire field of view once you turn these cancer cells into differentiated muscle fibers. And so we looked at this in multiple different cancer cell lines of the same type of cancer. This is, again, an alveolar cell, alveolar rhabdomyosarcoma. This has a fusion gene. If we knock the fusion gene out in these cancer cells, we don't see the large muscle fibers, but we do see almost complete differentiation of the cancer cells into differentiated muscle fibers. Now, the other type of rhabdomyosarcoma that has RAS mutations exhibits a similar type of phenotype. If we target the oncogene, we get differentiation of those those cancer cells into muscle fibers. But what's unique about all of these is each cell type is unique in its phenotype once we've disrupted that oncogene. Some of them produce those large muscle fibers that I showed at first with lots and lots of nuclei, and other ones are only have a single nuclei or a couple nuclei in them. And we think that this may be due from the original cell types from which that cancer was derived, or changes that occurred over time as cancer progressed in those cells. So we don't have a good idea of why the phenotype is different in these cells and why fusion is really important in some and not in other ones. But we do know that if we target the oncogenes that's driving that cancer, those cells are now transitioning into a myogenic state. So what that essentially tells us is those oncogenes are really important, or the landscape of the cancer cells are really important for not just driving progression of the proliferation of the cancer cells, but also blocking differentiation of those cells into muscle fibers. It's a really important check that we don't always think about, that in order for those cells to keep progressing, they need to stop differentiation. So how does this process work? This is something that we wanted to try to get our heads around. What is the mechanism behind how the transformation of these myoblast-like cells into a proliferative state? How would this work by blocking differentiation? And we got some evidence of this from large-scale drug screens that we performed actually before all this work, looking at over 40,000 different compounds one of the compounds that was able to induce differentiation of the cancer cells was HDAC and histone deacetylase inhibitors. And they were able to show some types of differentiation of these cancer cells, although not on the scale, obviously, of what we're able to do with CRISPR. And so this suggests that since there's not a high mutational burden in this cancer, that there may be an epigenetic signal that's helping to repress differentiation of those cells. So histone deacetylases, I just wanted to go over really quickly. They're essentially removing acetyl groups from histones and compacting the DNA. And this is a strong way to try to repress gene expression in genes such as tumor suppressors. Is common in cancer that these are overactivated. And so what we want to try to do is look at the whole entire family of histone deacetylases in this myogenic, or this myoblast-like cancer line. And so here's just an example of the different classes of histone deacetylases that are involved in humans. And we wanted to look at class 1 and 2 histone deacetylases. And so to try to get an idea of how they all worked, we essentially performed a arrayed CRISPR screen where we essentially did this on scale, where we looked at knocking out every single one of the histone deacetylases in class 1 and 2. And we did this in a unique approach where we tried to do it at a high enough efficiency where we can just deliver the CRISPR to the cells, and what the 90% of the cells will have the gene destroyed, and we can observe the phenotype directly from those cells. And in order to do that, we essentially target the gene at multiple locations, either 2 or 3 sites, to try to get that high efficiency. This was either done by transfecting the cells, these cancer cells, with plasmids expressing the CRISPR vectors or lentivirus vectors. And we essentially just let them grow for 7 days and then analyze their cell growth and their differentiation. So on the right, we have just showing that we're able to create high efficiency deletions in those genes, and then we're observing the phenotypes. And if we look across all of the class 1 and class 2 histone deacetylases, we observe that targeting of HDAC3 gave us that, caused the cells to have a massive amount of differentiation, similar to what we saw when we target the oncogene itself. Now, this was kind of an approach that we had to get around because we originally tried this by isolating individual colonies, and we could never get any colonies from HDAC3 because they would differentiate essentially. And we didn't know that at the time until we did this approach where we could do it at high enough efficiency. So to try to then get to the mechanism of this, we created an inducible cancer cell line where we could essentially treat them with a tamoxifen inducible Cas9 so that we can give them the drugs, and then the endogenous gene will be destroyed. And we essentially made one for HDAC3, and we can get the same phenotype where we target that gene with tamoxifen treatment. And you can see that we can get differentiation of those cells, but cells that are just expressing a scramble, treatment of tamoxifen does not induce differentiation. Now, one thing I can mention here is this differentiation doesn't occur with any serum starvation or anything. This is just full strength media. They'll differentiate on command, essentially. So the basic mechanism that we knew from previous work is that HDAC3, one of its roles is to function within a complex with NCOR, nuclear co-repressor 1 and 2. And essentially this complex sits on transcription factors and can block the downstream transcription of the downstream genes. And so we wanted to look and see, is the HDAC3 that we're involved in this phenotype, is this working through the NCOR complex? So in order to do that, we essentially targeted the NCOR genes. We targeted them individually, 1 and 2, and then in combination. What we did see is that they are redundant in function. So if we target one or the other, nothing happens or very little differentiation happens. But if we target both of them, we again get this massive differentiation of cancer cells into differentiated cells. That's similar to targeting HDAC3. So this led us to believe that this complex was probably involved because we could target both members and get the same phenotype, but we wanted to try to confirm that. So in order to do that, we did structure function studies where we essentially made overexpression constructs that don't have the CRISPR, that have the CRISPR target sites mutated in them. And that would allow us to essentially overexpress mutant forms of that of HDAC3, and then use our inducible system to destroy the endogenous form, while at the same time we are overexpressing different mutants to test their function. And so doing this, we created 3 different overexpression constructs. One of them was just the wild type HDAC3 to rescue the phenotype. The other one was a binding mutant that essentially was deleting the binding domain for NCOR 1 and 2 so that the complex could never form. But the HDAC3 was still catalytically active. And the other one was that had the binding domain, but was not catalytically active with a mutation in the actual catalytic domain. And what we saw is if we treated ourselves with lentivirus overexpressing those different constructs, we can see the treatment of the cells on the bottom left. That's our standard control treatment of the tamoxifen cells. Knocking out HDAC3 causes differentiation of the cancer cells. Overexpression of the wild type can rescue that phenotype even in the presence of tamoxifen. If we delete the binding domain, we then lose that rescue. So that means that the complex needs to form. And that what was interesting that we didn't expect at first because we didn't think about it, but overexpression of the catalytic domain actually caused differentiation in the cells that had the endogenous gene knocked out, as well as cells that had the endogenous gene functioning, because it's working essentially through a dominant negative inhibition of HDAC3. And that's what we kind of see on the far right panel in NNO there. So what this essentially told us was that this complex was important for blocking myogenic differentiation in these cancer cells, that it needed to form, and that the catalytic activity of HDAC3 also needed to be there. So to try to get further down into the mechanism of how this complex was working, we did essentially pull down to show that the complex is binding with MyoD and E2A, and binding to that complex. Part of the repressor complex is actually binding to transcription factors. And so this was the transcription factors that we were looking at and asking, could it possibly inhibit MyoD? When we look at RNA sequencing of those cells, we could see that the MyoD target genes are upregulated after we target HDAC3. We target HDAC3 and we see an upregulation of the MyoD1 target genes. We can see that if we take and look at the binding using CHIP assays, we can see that HDAC3 binding to, if we knock out MyoD1 in the cancer cells, HDAC3 binding to the location, the promoter region of specific, the MyoG target genes. So essentially HDAC3, if we target MyoD, because the complex we hypothesize that it's binding to MyoD at the promoter of its target genes. If we knock out MyoD, we see a loss of HDAC3 binding at those target sites. If we look at the acetylation of the genes themselves, there's an increase in histone acetylation at those target sites when we disrupt HDAC3. So HDAC3 is going to deacetylate the promoter regions of those genes. So if we disrupt it, now we see an increase in the acetylation of MyoD1 target genes. And then if we knock out MyoD, E2A, CBP, and EP300, we can essentially reduce the ability of those cells to undergo differentiation. Now, this is kind of a given because they're involved in the myogenic process. We didn't necessarily see that. While it was significant with MIF5, it was more important for MyoD1. But together, we believe that this complex is sitting on the MyoD and acting as an inhibitor. So the big kind of model that we're trying to work on and see how this could all play together is that this repressor complex is actually important for controlling the function of MyoG. So even though it's expressed and working, this epigenetic complex is sitting on those transcription factors and blocking their activation of downstream genes. And that the key point is that in order to trigger myogenesis, we need the removal of this repressor complex. Now, we did look at some primary cultures, and we do see a very transient presence of HDAC3 in the nucleus of the cells right during the differentiation that we, but it's so transient and quick. I think that is part of the reason why it's difficult to study in cells that aren't, that are normal primary cells, is because this inhibitor or this repressive complex may be a very short-lived event. But in the cancer cells where we have essentially a proliferating myoblast, we can look at this process in more detail. And we believe that in the cancer cells, it's the mechanism of this complex binding to the transcription factors.
4 minutes left.
Okay. That this transcription, this repressor complex is somehow dysregulated in its ability to come off of the transcript of MyoD to activate differentiation. And so we looked at the expression of the NCORs and the HDAC3s, and they're not differentially regulated in the cancer cells compared to, we don't see any upregulation of those genes or anything like that. And so the mechanism is not dysregulation of the complex itself. It's dysregulation of some kind of activating event that would cause it to be removed and therefore allow myogenesis to proceed. And so this is kind of how we're developing the model for how this complex may play a role in that trigger of differentiation in myogenic cells. So this is just my acknowledgement slide. This is from I work with Eleanor Chen and at the University of Washington, and I'm happy to take any questions. I went through it fast so we can catch up on time.
Zipporah, you have a question?
I can only tell the audience that Michael Phelps did extremely well. He once was a postdoc in my lab, and I am extremely impressed. And he now has his own lab in Washington State University. He's a magnet for students, and he works on fish, and I'm advertising for him, of course. And thank you, Mike. Thank you, Mike, for a wonderful talk.